Good evening or good afternoon, everybody, and welcome to the 4th annual Alan Turing Lecture. Of course, it should have been the 5th annual Alan Turing Lecture, but very sadly, due to the pandemic, no lecture took place last year, and indeed our lecturer this year would have come last year to lecture. Many of you know me, but for those who don't, I'm Professor Michael Proctor. I'm the Provost of King's College, and I'm Chair of the Alan Turing Committee. King's has always prided itself on being somewhere that gives creative and curious minds the opportunity to think differently, to question the status quo, and to excel. It's hard to think of anybody, alumni or fellows, who've passed through this place, who exemplify that more than Alan Turing. In his lifetime, his work had enormous impact. And as we know, many of his ideas and innovations were decades ahead of their time. With the Alan Turing at King's program, we want to build on this spirit of inquiry and urge to interrogate the world. And we encourage and support the bright minds who could be the Turings of tomorrow. In particular, we host each year a lecture by a distinguished academic on either Turing the man or on a topic related to his scientific work. King's and Cambridge has always had extraordinary power to convene the best people and shape new ideas. And I'm delighted that we're demonstrating this further today with our distinguished lecturer and the discussion that will follow. I'd now like to introduce the panelists, and very pleased to see them now on screen. So this year's lecture is Modeling Pattern Formation in Developmental Biology, and it's going to be given by Professor Philip Maini from the University of Oxford. Professor Maini is an FRS and is a distinguished mathematical biologist. He's the head of the Wolfson Center for Mathematical Biology at the Mathematical Institute in Oxford. In his research, through sophisticated mathematical modeling, he's elucidated many of the mechanisms behind pattern selection and morphogenesis in biology and medicine, a subject of great interest to Turing, who was one of the first to understand the biochemical basis of pattern formation in biology. Following the lecture, King's Fellow Dr. Ronojoy Adhikari will also join the session to take questions from our live online audience for Professor Maini, not our 5 online audience. It's a very large audience of over 100 people today. Ronojoy Adhikari is a lecturer in applied maths at the Department of Applied Maths and Theoretical Physics here in Cambridge. His interests are in the synthesizing principles from geometry, mechanics, and probability to model physical and biological phenomena. And so I will now hand over to Philip for the lecture. Thank you, Philip.
Thank you. So hopefully I will share the screen now. Okay, so I'd like to begin by thanking the college for inviting me to give this talk. I feel very honored to be asked to give this talk, and I'd also like to thank the Provost for a very kind introduction. So what I will do in this talk is I will talk a little bit about Turing's work in developmental biology. I will then talk a little bit about some of the work that I was involved in, going back almost like 20, 30 years ago. And then right towards the end, I'll mention a bit of work which is not on Turing pattern formation, but it's still in developmental biology. I'll mention a bit about that at the end. So what we are looking at is mathematical models. So a natural question to ask is, what is a model? So to an experimentalist, these are models. So a Petri dish or a mouse. So you're trying to understand what goes on, say, in the human, in developmental biology, and you can't do lots of experiments on people. So you do experiments in a Petri dish or in a mouse, and you hope that you can relate the results from those experiments to what's happening in the human. And in fact, the best definition of model that I've seen is actually from Alan Turing himself. So in his paper, The Chemical Basis of Morphogenesis, he talks about his model. He says this model will be a simplification and an idealization, and consequently a falsification. It is to be hoped that the features retained for discussion are those of greatest importance in the present state of knowledge. And I should say I've bold-faced these things here. Here's the original down here. And a little bit more succinct definition is from the statistician George Box, talking about statistical models. He says all models are wrong, but some are useful. And so basically, we all do models, and we have different modeling approaches ranging from experimental to statistical to mechanistic. And these have different levels of abstraction and different levels of understanding and different levels of tractability. And one of the key aims in the field at the minute is how to bring all of these together so that we can learn from the strengths of each one of them. So Turing wanted to know how patterns form. So here is a cheetah in a cheetah reserve, and it's sleeping. And you can see here the obvious patterns are these spots. But of course, there's also all the structure of the limbs, the face, etc. There's lots of pattern here. So he wanted to know how we got these coat markings, and for example, how you got trees branching. Because if you look at this tree here, as it's growing at this level, it's circular. So you've got a circular symmetry. And then that symmetry gets broken as it branches. So the question is, how does that arise? And so what he said, it is suggested that a system of chemical substances, and he called them morphogens, reacting together and diffusing through a tissue is adequate to account for the main phenomena of morphogenesis. So for example, in the case of the tree, what would happen is the idea is that you've got a growth hormone, which is distributed with circular symmetry, and then that symmetry becomes destabilized, and the symmetry is broken, and you get local maxima, and then that causes the tree to branch. And so, as mentioned, he called these morphogens because they give rise to form, and that basically they form a pre-pattern to which cells respond. Okay, but now if we go back and think, what did he actually say here? He said that diffusion is key. And that doesn't seem right, because diffusion wipes out pattern. And we all know that. For example, here you put a drop of different colored liquid into the water here, and it diffuses. And eventually, because diffusion is random movement, there's no preferred position, it's going to wipe out structure. And mathematically, we can prove that. So here is a reaction diffusion equation. So here's diffusion, here is reaction. And so I'm just putting some equations up so mathematicians can see this, but I'm going to go through it very quickly so that we don't sort of alienate the non-mathematicians in the audience. And so the reaction term here would be production and degradation of the chemical. And so if we were to take something called a spatially uniform steady state, so we have a value of the chemical C, which doesn't change in space, doesn't change in time, and is basically at equilibrium. So it's an equilibrium value. And then let's assume that we perturb that slightly, and we want to find out if we perturb it, will it move away from equilibrium, or will it go back to equilibrium? You can think about this, for example, if you have a pendulum and the pendulum is lying down, if you move it slightly from that steady state, it will come back to that steady state. So that steady state is stable. So what we want to do is find out if we make small deviations from equilibrium, do they grow or do they decay? And if the deviation is small, we can linearize the system. And then we can solve using Fourier series. And so what you see here is if the diffusion coefficient is very small, then we have what could possibly be exponentially growing if f dashed of c is bigger than 0. So that means that if diffusion is very small, perturbations will grow exponentially. So the system is unstable. The equilibrium is unstable. But if you made diffusion sufficiently large, these will all be negative. So diffusion stabilizes structure. So we can take something that is unstable in the absence of diffusion, and diffusion stabilizes it. And Turing said the exact opposite. He said, I'm going to take something that is stable in the absence of diffusion, but becomes unstable in the presence of diffusion. And crucially, he considered a system of two chemicals, not one, but two chemicals. And in fact, he considered them on a ring. And so the idea here would be that you had, for instance, a cell here and a cell here, and then the amount of chemical here was XR, and then XR minus 1, XR plus 1, and then you looked at the flow between these, which depended on the difference. And then if you write down this equation here, what you can see here is essentially you've got a discretized version of the Laplacian. And more generally, this would be the form that the system now looks, where when we go to more than one variable, in dependent variable, we just put vectors along everything. And then he did linear stability analysis, essentially. And essentially, he came up with these conditions. So where f and g are the different functions, and u and v are the different components of the chemical. So what these were the conditions for the steady state to be stable in the absence of diffusion, and these are the conditions for diffusion to drive it unstable. So the crucial thing here is going to more than one species. And now you can see here that this means that not every reaction diffusion system can give rise to these patterns. It must satisfy these conditions. So one thing it does, if you look at, for instance, here, these df by du plus dg by dv must be less than 0, but this combination must be bigger than 0. It means that these two must have different signs. And if these two have different signs, it means that these two must have different signs. And so basically, you have this setup here. Either you can have this setup for these functions or this setup. And now let's look at this one, the second one. So the second one says that df by du is positive. So that means if I increase u, I increase f. And f is the production of u. So it means u increases its own production. So it activates itself, or it's autocatalytic. And look at this one. dg by du is positive. So that means if I increase u, I increase g. G is the production of v, so it means u increases the production of v. So u activates v. But what does v do in return for this nice help? Look at df by dv. df by dv is negative, which means if I increase v, I decrease the production of u. So v inhibits u. So you've got what's called an activator-inhibitor system. And then if this fu is positive, and the GV is negative, then you look at here, well, obviously D2 and D1 can't be equal to each other because they're both positive, so they're equal to each other. These would contradict, these two inequalities would contradict each other. So what we find out is that D2 has to be bigger than D1. So we've got that the inhibitor diffuses faster than the activator. And this is the idea of short-range activation, long-range inhibition. So the idea would be here, you start off with this uniform steady state, you perturb it slightly, and it goes unstable due to diffusion, and that sets up a chemical pre-pattern. So here's the cells, and the cells then have different fates responding to these different morphogen values. So beforehand, people like Waddington had talked about evocators and said there's some signal or something that causes cell fate to change. And Turing said that something is a chemical or chemicals, and he called those morphogens. Now, here's a very silly example I dreamt up of short-range activation, long-range inhibition. Let's consider children. Well, we all know the job of children is to make lots of noise. They create noise and they activate themselves. So they encourage each other to make more and more noise. So they are, that's autocatalysis. And what they do is they activate the adults to inhibit them. So the adults go, shush, keep quiet. So imagine a situation where you have a sort of, if this was balanced, there would be a uniform small level of noise. Okay, noise here, I mean sound, I don't mean mathematical noise. Okay, so that you could have a uniform state of noise where if it gets a bit too noisy, the adults say shush, and you go back down to this equilibrium. If it gets too quiet, well, we know children can't stand silence, they'll create noise again, and you'll go back up to this equilibrium. So we have this equilibrium that's stable in the absence of movement. Now suppose everybody starts to move. Well, the adults move faster than children, so the inhibitor diffuses faster than the activator, and so soon you'll find pockets where there are many more children than there are adults, and the noise can grow. And this will be surrounded by areas where there are lots of adults and maybe only one child, and if the child opens its mouth, the adults will go shush, so there'll be no noise. So you get a spatial pattern. And crucially here, what we see is that our intuition that diffusion is stabilizing is actually only for the singular case where only one species is diffusing. As soon as you have more than one species diffusing, diffusion can drive instability. So that's the intuition or the fantastic new insight that came from Turing. And then here's a simulation that Turing did of his model, and you can see you get these nice patterns. And just a quick aside here, I've emphasized how you need 2 or more species. That's only if we have diffusion. If you have more complicated transport mechanism, for example, like Cahn-Hilliard, which can be considered as a nonlinear diffusion, you can get a pattern with only one species. And this model has been used a lot in phase separation and segmentation and stuff. But I'm just dealing with simple diffusion here. Okay, so now the question is, we get the pattern, what does the pattern look like? Well, it turns out the pattern respond to eigenfunctions of the Laplacian. you write the solution as separating as a separable solution, and it turns out you're essentially taking a Fourier transform of the solution. And so the spatial aspect of the chemical will be an eigenfunction of the Laplacian. So one thing we know with eigenfunctions of the Laplacian is that, for instance, in one dimension, if the domain is very small, there'll be no eigenfunction that will fit in the domain. So there'll be no pattern. As the domain increases, the pattern will become more complicated. And basically the reason for that is when you do the analysis, you find out that the patterns, for example, in this particular example I'm looking at with zero flux, zero flow out of the boundary, you get solutions of the form cos n pi x over L, where n is an integer, and n squared pi squared over L squared has to be bounded by these two values that are determined by the parameters of the system. So that comes from looking at the dispersion relation. But the key take-home message here is you can see that if L was very, very small, then for any value of n non-zero, this would not fit in here, so I get no pattern. As L gets larger, I have to increase n to keep fitting in this interval. So the pattern becomes more complicated. If you go to 2 dimensions, then you find you've now got 2 scales, the sort of length and the breadth, and now you have to satisfy this condition. And so what that tells you, for example, if Ly was very, very small, so you had a very thin strip, then M would have to be 0. So you just get a pattern in the x direction. But if the Lx and Ly are both large, then both n and m are non-zero, and then you get patterns that vary in space and in time. And that's just reading here that if you've got a strip, you'll get stripes. But if you've got a broad domain, you'll get spots. And so now just some examples of these reaction terms. Here's a very famous one, Germ-Meinhardt model. And this is just a phenomenological model. Meinhardt realized independently of Turing, he didn't know about Turing's work, he realized you had to have this activator-inhibitor system. So he simply chose F and G so that here U is activating itself and it's activating V, and V is inhibiting U. Here's an example of an empirical model where this is for uric acid and oxygen interacting, and here what happened here was just you fit this to data and you get these F and Gs. Or you can get the F and Gs from a more mechanistic by actually writing down chemical reactions and using things like the law of mass action. So from a biological point of view, these Ks would, like our growth rates, reaction rates, and death rates, they would eventually be linked down to the genetics of the system. They would contribute to these different values of these parameters. Okay, so this is all asking the question, what would happen if I just did a small perturbation from the equilibrium, and we show that under certain conditions, that perturbation will start to grow. Then our analysis becomes invalid because we assumed there was a small deviation. As soon as that deviation gets too big, our analysis breaks down. And this is still an open question. How do we analyze these patterns for the full nonlinear system? You can do some weakly nonlinear analysis. There's a lot of numerical simulation, bifurcation analysis, in fact, even group theory ideas, semi-group theory ideas of how do these patterns really behave for the full model. It turns out that in many cases, linear analysis is a pretty good predictor of patterns. So here's some applications, and Jim Murray, who I was very lucky, he was my supervisor, one of the pioneers in the applications of Turing models. And so what we have here, so this is numerically solving the Turing model on a domain that looks like an animal coat. And what's happening here is the domain is increasing in size as we move along, but we've rescaled it so it all fits on the page. So what you see here, linear analysis predicts when there's small domain, you'll see no pattern. And this is a numerical simulation of the full model, and it's correct. As you increase the size of the domain, you get pattern. The patterns become more complicated, but now you see here the linear analysis breaks down. Okay, so now the question is there an animal that looks like this? And this is actually a true goat. Is there an animal that looks like this? This is my favorite. I love cattle. Here's a belted Galloway. Okay, and now you see here, again, here's numerical simulations of the Turing model. And you can see here, this looks more two-dimensional, so you get spots. And as you go down, it becomes one-dimensional, just stripes. Likewise here, just stripes. In fact, maybe at this point, I should mention that this tie I'm wearing is actually a Turing pattern generated by my former student, Thomas Woolley, who's now faculty at Cardiff, and he's got the matching waistcoat of this tie. And here's some, and what you can see here is, if you look at these limbs here, you can see that this is actually real limbs, not numerical simulations. The pattern gets more complicated as the domain widens. It happens in all of these. And so here's another example. You see here where the domain is more two-dimensional, you get spots. And then when it becomes narrower, like one dimension, you get stripes. And this particular model, this particular paper here, Developmental Constraint, comes from two experimentalists who simply looked at lots of pictures of patterns and concluded that you're more likely to have spots going to stripes than stripes going to spots. So that's data-driven modeling. And then Oster and Murray, the two mathematical modelers, using the ideas of Turing, give a mechanistic understanding of why you have spots and stripes more likely than stripes going to spots. Okay, because the domain here is more two-dimensional, so to speak. So you could have spots here, one-dimensional, you could have stripes. But if the system is set up so that in the two-dimensional domain, the most complicated pattern you can get is stripes, then on the one-dimensional domain, you can't get a more complicated pattern. So the pattern is either going to be stripes or nothing. Okay, here's another example of that developmental constraint. Football shirts. You see here stripes, and then on the arms, the pattern is less developed. Here, in all of these, the pattern on the sleeves is not more complicated than the pattern on the body. It's either as complicated or less complicated. Premier League football shirts from the season 2016-2017. What we see here in none of these is the pattern on the sleeves more complicated than the pattern on the body. Rugby shirts, same thing. Okay, so what we've got here is a mathematical model for the coat markings of football players, be they association football or be they rugby football players. So being serious now, this raises a very important point. Just because the model agrees with observations doesn't mean it's the model, right? The Turing model was not used to generate David Beckham's shirt. We have to do experiments. Well, experiments would say, if you were to change the size of the domain, for example, the limb part that forms digits, if you were to make that smaller, would you see fewer digits? Or would you see the same number of digits, but just smaller? Well, the Turing model predicts you should see fewer digits. And this is what happens. So one of these is salamander and one is frog. You reduce the domain size, you get fewer digits. What would happen if you increase the domain size? Would you get the same number of digits, only they'd be fatter? Or would you get more digits? The Turing model predicts you will get more digits. And here's the experiment. And the chick, normally you'd have 3 digits, but now you get 6. You double the domain length, you got 6 digits. Now here's an experiment by Walpert and Hornbrook, where they did double anterior limbs. So they took this part of a limb in a chick, threw it away, took this part of a donor and stuck it in there. So double anterior limb. Now the Turing model predicts you haven't changed the size, so you should get normal structure. And you don't. You get 2 humeri forming instead of 1. So this contradicts the model. So we looked at this quite a long time ago, and we built on work that Othmer and Pate had done when they were looking for scale invariance in another example. And they used the idea that what would happen if you had a spatially varying diffusion coefficient. So what we did was we said, well, suppose there's another chemical, and suppose it forms a simple gradient. And suppose that chemical controls the gap junction permeability of cells. So that means it affects the diffusion. And let's assume that it affects the diffusion of the faster chemical, the inhibitor, and not the activator. Then what would happen? Well, here's your humerus in the normal, and then you do the experiment and you get 2 humeri. So we submitted this paper and we thought, well, there's no evidence that there is this diffusion spatially gap junction permeability across the limb. So, you know, we're a bit worried. People might just say, oh, you're just making up some mathematical toy to get the answer that you want. While this paper was in review, which takes a very long time for mathematical papers, this paper here came out in which they showed that retinoic acid modulates gap junction permeability. And crucially, there's a gradient of retinoic acid across the limb. Exactly what we were saying we thought would happen. And in fact, so this brings together the Walpert's classical positional information model, where he said there's a gradient and cells respond to different levels of the gradient. And it brings this together with the Turing model. And of course, Turing already knew this, because Turing said most of an organism, most of the time, is developing from one pattern into another, rather than from homogeneity into a pattern. And so other mechanisms that give rise to patterns that are eigenfunctions of the Laplacian will also give Turing patterns. So here's a very nice example again from Jim Murray's book, where we know that a vibrating plate, the modes of vibration are eigenfunctions of the Laplacian. So they took a vibrating plate that looked like an animal coat marking, they vibrated it, they colored the displacement, and you get animal coat markings. Here's some counterexamples. Striped body, spotted tail. No pattern, but pattern here. So what's going on here? Well, what's going on here is that maybe other parameters are changing, because this idea of spotted animal with stripy tail, striped animal can't have spotted tail, you're assuming that the biochemistry is not changing as you move around the animal. But if the biochemistry is changing, then that doesn't work. That constraint doesn't hold. Here's another example of a counterexample: striped body, spotted arms. And here's another beautiful work by Hans Meinhardt, who looked at seashells. And what he did here is slightly more complicated reaction diffusion model. And here he took the reaction diffusion model on a two-dimensional growing domain and then projected it onto a growing shell. And you get this. And now, since that work was done, which is 30 years ago, there's a lot of work done on actually, with the advances of computation, on solving these equations on evolving surfaces. And that's a big area of research at the minute. What do these patterns look like? But he's got some very nice pictures here: real life Turing, real life Turing, etc. And of course, they can also apply to ecology. Patterns, Turing patterns, have been applied in ecology. So do Turing patterns exist? So what's happened is, in biology, many potential activator-inhibitor morphogen pairs have been identified. But really showing that these are Turing, there's still a step that has to be done. But in chemistry, they've been shown to exist. And so these are patterns, Turing patterns, that arise in a chemical reaction. And that chemical reaction is the reaction of iodide with chloride. And in fact, what happens here is the chloride also, or the iodide, combines with starch. And so you end up getting a 3-species equation. And you can do, and these people, Langell and Epstein, did the analysis, but you end up getting, they show that you can get these patterns, complicated patterns, labyrinthine patterns, that are related to, that are showing to exhibit in the chemical species. Now we have, here's a very classical model of normally what happens, I mentioned the thing that as the domain grows, the pattern becomes more complicated. But a key thing is that cells can only respond to external cues and change their fate for a certain period of time, window of time. So for example, when our cells decide to form our head, and then as our head grows, we don't form another head because simply we can't respond and change our fate. But what Kondo and Asai found was if you looked at the angelfish, Homocanthus, as it grew, and these stripes, when they became twice the size, twice the spacing that they would normally have, another stripe appeared, just like the Turing model would predict. And in fact, sometimes this second stripe appears by a zipper mechanism, and they showed that you could get that from a Turing model. So it's quite complicated how these secondary stripes can arise, but they showed that the Turing model could exhibit these stripes. Now, if you go back and look at this, what you see here is that these new stripes are thinner than the previous stripes. That's not predicted by the Turing model. And so what we did was we made the assumption was, let's suppose that the pattern we see is not the pattern in chemical, but is actually a cell pattern. So suppose the cells move in response to the Turing pre-pattern. So they don't just sit there and change fate, they actually move. And they move via the process of chemotaxis, which is movement up a chemical gradient. And then we solve these in a growing domain. You have to put advection in and various other things. But this gives you thick and thin stripes. And then if you look at more complicated problems where you have the cells responding to both chemical cues, you can get all sorts of patterns. So what we're showing here is that if you couple pattern generators, and remember Turing said it's not going from homogeneity to pattern, it's going from one pattern to another pattern. So what we're doing here is we're doing exactly that. We're having this system giving a Turing pattern, and then the cells responding to that Turing pattern to form their own pattern. And you can get these complicated patterns. And so this is still a very, very active field. And in fact, we are presently editing a special issue of the Philosophical Transactions, on key aspects or the new challenges in the Turing model. So what I will do for the last 10 minutes is very briefly describe some very recent work that we're doing, which asks the question, well, what we were doing in the Turing model is we're saying once the cells are where they're supposed to be, how do they do what they're supposed to do? But a natural question to ask is, how did the cells get there in the first place? Where did they come from? And a lot of the cells I've spoken about come from something called the Neural Crest. And this is work, I should mention, has been going on for 10 years with experimental collaborators at the Stowers Institute for Medical Research and with colleagues and graduate students at Oxford. So graduate students Louise, Linus, and Raza, my colleague Ruth Baker, and Paul Kalesa, and Rebecca McLennan at the Stowers. So basically what happens, this is a chick, this is the head, this is the tail down here. These neural crest cells emerge from the neural tube and they go to something called the branchial arches. And we were looking at these ones here that go from rhombomer 4 to branchial arch 2, and they form the lower jaw. Now, why study this? Well, there are many, many birth defects occur as a result of this movement not happening properly. And also these particular neural crest cells are very similar to some of the most aggressive cancers. So these particular neural crest cells have very little proliferation, and you can easily show that if the cells just moved randomly, they would not be able to progress from rhombomer 4 to branchial arch 2 in the correct time scale. It would take them too long. So a model that was proposed was that there was a chemoattractant. It was known that a chemoattractant was being produced by the tissue. And so the idea would be, as the cells come out of the neural tube and they consume the chemoattractant, that would create a gradient, and that would give them a biased random walk. So they wouldn't, and therefore they could move and invade the tissue more quickly. So what we did, again, I'm not going into the details since we're running out of time, but this is basically just a reaction diffusion system for the VEGF on a growing domain, and this is internalization of the VEGF by the cells. And this is experimentally determined growth of the tissue. So we consider the tissue as a rectangle growing. And we assume that we take various boundary conditions, and we assume, because there's a small number of cells and stochasticity is important, we assume that the cells stochastically sense the gradient by sticking out filopodia, and they move up the chemical gradient. So I'm rushing through this, but basically, I'm happy to go into details in the question session. And so here we have, here's the domain growing, here's the VEGF, and look at this, it's brilliant. The cells are coming in, they're sensing the VEGF, and they're moving up the gradient of the VEGF, and we're getting a really, really lovely invasion, just like the verbal model said we would get. So this is not working. Oh, these cells got left behind. That's a pity. So the reason why they got left behind is this gradient has got localized. So then we thought, well, okay, then, suppose these cells responded to the cells in front of them because they've got no chemical to respond to. Suppose they stuck to the cells in front of them, then what would happen? And I won't show the video, but you get movement. You get nice streams forming. So we went back to our biologists and we said, are all these cells the same? Because we think there might be a difference between the cells at the front and the cells at the back. And they said, well, we don't know, but we'll find out. So they did a gene analysis, loads of bioinformatics, and they found that the cells at the front were different to the cells at the back. And they were absolutely delighted when they saw the cells at the back upregulated cadherin 11. And we said to them, why are you so excited about that? And they said, well, cadherin 11 is to do with stickiness. So the cells at the back are stickier, just like you predicted. And we went, oh, right, great. So then we did a lot of experiments comparing the model with real experiments. A lot of the times we agreed with the experiment. I always think you learn more when you don't agree with the experiment. And here's one thing where the model didn't agree. So here we take followers and we implant them to the front of the domain. And the model, mathematical model, predicts, since these are followers, they don't respond to the chemical, they'll just be advected by growth. What's observed experimentally is they actually actively move. So they change their phenotype, they become leaders. So the obvious way to check that hypothesis is, let's now put VEGF along the stream. And then what the hypothesis would say is that as the cells are moving along, the followers, when they see this new VEGF, they become leaders, and then they move up, and the stream should split. And that's what happens. And so we've done a lot of these experiments back and forth, but now I just want to show you one last example here. I very quickly didn't mention that in our new model, look at these leaders. They've got too far away. They're too far away from the followers. They're not going to be any use in getting the stream. Moreover, why we just didn't allow the cells to move out of this corridor. So there must be a chemical that's stopping the cells from moving out of the corridor and stopping these cells from moving too fast. Otherwise, the model's not going to work. And then they found the chemical, or not the chemical. What they noticed was right next to rhombomer 4, something called DAN. The gene DAN is upregulated. Now, DAN is a BMP inhibitor. It slows down motion. So as the leaders come out of here, they're slowed down by the DAN. So they can't move fast. And as they move through here, the DAN disappears. And so now the followers can move at normal speed, and so you've got the coherent movement. And here's just the experiments to show that DAN, if you put stripes of DAN, the cells don't move up on DAN, they move away from DAN. I mentioned that one of the reasons for looking at cranial neural crest was the relationship to cancer. So here's melanoma cells. So now, obviously, what we're going to do, we're going to confront melanoma cells by DAN, and they move away. So they still move a bit, but statistically significant decrease in their movement. So what we've seen here with this simple mathematical model linked with experiment is we've been able to actually unearth quite a lot of new biology. But this has also given us new mathematics, because which we haven't discovered yet, we're still trying to do this, we're now trying to write down a full mathematical investigation of this, where you've got this growing domain, you've got these cells, they're changing type, how they change their type depends on the environment. That leads to quite a complicated mathematical model that we're still trying to derive. So I'll end there with the conclusion, and I hope what I've illustrated here is that biology leads to new mathematics. We saw Turing developing the reaction diffusion system to try and explain something in biology. That's a classical example of it. And in turn, mathematics leads to new biology. For example, in the case of Turing, he identified there should be morphogens. People have found morphogens. They have found morphogen pairs. There's a huge amount of investigation now on, are these behaving exactly like Turing predicted? And then our small contribution towards the end in terms of Neural Crest was identifying different phenotypes, identifying phenotypic switching, but also that's going to lead to new mathematics of how do you model this phenotypic switching, which will be in a sort of state-structured model. So what I will do then is I will stop there. Thank you for your attention and happy to take any questions.
Well, let me thank Professor Maini for that very, very nice talk. We already have some questions in the question answer box, but I'll encourage the audience to add more questions. I'll start the question answer session with a question from our provost. And Mike asks, how has the advent of high-speed computing changed the way in which models are constructed?
Well, very much so, because like I mentioned, the Meinhardt work, where what he did was he actually took, he solved the equations on a flat, two-dimensional flat surface, and then he projected that onto the curvature of the growing shell. But now that we've got advent in high-speed computing, we can actually sort of pose the model using the Laplace-Beltrami operator, etc., on the curved surface. And then we can ask the question, okay, now we've got this curved surface and it's growing. Does the pattern form in the way that that we observe experimentally, or just by looking? And what is the effect of curvature on growth? There's quite a lot of work being done on that. And then also it allows, what Turing said about patterns are forming one after another, and there's this integration of patterns pattern generators are integrated. We can do more of that. We can also determine parameter spaces in which these patterns form, and to find, are they robust? Because this is one of the other issues with the Turing model, that sometimes if you change the parameters a little bit, you can get a different pattern. So those are a lot of issues that we can address now because we've got much better computing power. So it's revolutionized how we can analyze these models numerically. And that, of course, leads to then analytic questions of, can we do an analytic theory to explain what we see numerically?
Right. So would it be fair to say that the aspect of geometry, which perhaps Turing had neglected, we can now address through the...
Yeah, and I should say, actually, I mean, Turing did solve his problem on a sphere. He was looking at gastrulation, so he did look at a sphere. So I'm pretty sure that he knew that geometry was going to play a role in it. But we can look at that now with computing.
Okay, so there's another question from Professor Herbert Huppert, and he asks, what if the diffusion coefficient is spatially dependent and perhaps also dependent on the concentration? And I'll quote him, or yoicks time.
Or what, sorry? Or
no, that was just quoting him, or time.
Right. Yeah, so the thing is that, again, what we did in that paper with Hans Othmer and Kevin Painter was we sort of had it depending spatially through another chemical. And in some sense, that gives you an isotropic diffusion. And so one thing I didn't mention was that a lot of the two-dimensional patterns in biology actually form behind a front, a moving front, leaves the patterns behind. And what we think is happening is this could be spatial dependence of diffusion or of some of the chemicals. And that as the embryo is maturing, these chemicals or these parameter values are changing, and therefore the system's evolving. And again, this relates actually to the previous question. We can now study this by doing lots of numerical simulations, because to try and do this analytically is very, very difficult. So numerically, there's a lot of work going on in looking at this. And in fact, our special issue that we're putting together is going to showcase some of those. We've got lots of fantastic authors who are going to showcase lots. So this is absolutely a highly topical and highly relevant question that people are looking at.
I'll continue with the question and answers. So there's a question from Dermot Turing, and this is to do with the historical development. So he asks, why do you think the Wolpert 1969 model became the classical model for development, but Turing's 1952 model has only been explored more recently? The chronology seems curious.
Yeah, that's absolutely, I don't know the answer to that, because the Turing model, it sort of lay undiscovered almost. And then Wolpert, because Wolpert himself was a developmental biologist, so it was very, when he came up with this theory, he was able to do experiments, he was able to sort of propagate the idea of the of positional information. And then it was only when Meinhardt came along in the early 70s, and Meinhardt, as far as I understand, didn't actually know about Turing's work. And he developed this idea of activator inhibitor sort of independently. And then, because he was also working with developmental biologists, it was only then that the Turing model started to come to light. Even though it was, as Dermot mentioned, like 2 decades before Wolpert's positional information idea. And now I should say people like James Sharp, for example, are doing a lot of work on showing that it's probably you've got to combine the two, which again, very new when he said, you're developing from one pattern to another. He'd already anticipated that.
Right. Now, there are a couple of questions on evolution, and I think the evolution of interaction with Turing patterns, and I'll read both of them out because I think they're addressed together. So there's a question from Maria Gutierrez. She asks, does evolution in the Darwinian sense play any role in Turing patterns and the existing models for these? So that's the first question.
Yep.
And there's a question from John Bird. He asks, how did this complicated biology come to exist through evolution? I'm thinking there must have been a huge quantity of dead ends that got discarded during evolution.
Yeah, so the thing is that, this is also an whole, we always sort of skirt over the parameter values. I mentioned them briefly. Remember that for these patterns to form, the functions have to satisfy these inequalities. And then when you write out those inequalities, they will come in terms of the parameters. And so what that's telling you is that the parameters of the system must satisfy certain conditions, and the parameters eventually are related to the genetics of the system. And so what you might sort of think would happen is that maybe the patterns that that are selected are then propagated. So those genes that give rise to those parameter values get propagated. Because if you think about it, people sort of say that biology or the world is so amazing, so much pattern. And I say, no, the world is actually quite boring. If you look at all those limbs I showed, whether it be a human, whether it be a dog, whether it be a chick, is all the same. 1, 2, 3. You don't see 3, 1, 5, 6. And again, this is consistent with the Turing idea that as you expand and the domain gets larger, you're getting more structure forming. So there is the question of how do you move into the parameter regime that gives you these patterns? And then the other question about the very many dead ends. I mean, probably that's right. I mean, there must be, because, if you did have the structure of, 2, 1, 3, 5, 6, that probably wasn't very functional. And so your genes wouldn't get passed on. So there's the question of, if there's anything like that, do they get aborted early on? Or is there, I mean, at what level do you see the selection? And that, is something that I don't know because I'm not an evolutionary biologist. But I just can't help but think that the fact that we all exist means that there must be a way of looking at how we form where we think, oh, that's obvious that should have happened, rather than it be so complicated that so many things have to be just right for us to exist. But we just don't know what the principles are at the minute. That's what I think.
Yeah, so I think that really answers Tim Griffin's question, can we start to understand the interaction with natural selection? I think.
Yeah, so I think there would be the case of then, since this pattern information stuff doesn't really look at functional aspects, so that would be the feedback, that if something is functional, then that would feed back, so those parameters propagate in the system, whereas if it's not functional, then the parameters that gave rise to that structure would not propagate. That's what I would say would happen.
So I think there are a couple of questions on how Turing patterns relate to things beyond biology. So Ebu Versi asks, does the mathematical model underlie our perceptions of aesthetics vis-a-vis fashion? For example, football shirts.
Well, that's what I wondered. So when I gave a talk like this a few years back, I did raise that. Is it the case that subconsciously we develop these football shirts because we see patterns? We hardly ever see these things where as the structure narrows, suddenly you get more patterns. And I did wonder about that, but I've not investigated to see, whether people have looked at that from a psychological aspect. But I'm glad that somebody else has raised that, because I thought maybe I'm just being a bit strange by raising that idea.
Right. So there's another question from Richard Robinson, and he asks, can you say a word as to whether these models can be applied to fingerprint formation and whether the changes in the dermatoglyphics in, for example, Down syndrome tells us anything about the cell behavior in this condition?
So I don't know anything about Down syndrome, we'll have to say, but what I will say is that I know that, like Jim Murray, he did some work on fingerprint patterns using a different model from Turing. It was a more mechanical model. But so some people have done work on fingerprint patterns, but I haven't followed them. Of course, the issue of the fingerprint patterns then would be that why are they all so different? Whereas these types of models would sort of say, well, they sort of are roughly speaking the same, but there's differences from one to another. People have looked at that, but I don't know in how much detail they've looked at it. I'll have to say.
So let me come back to a slightly more technical question. Sorry, I'll just revise. There's another question which has come in, which probably is better suited to the flow of questions. So Barney Salsby asks, has pattern formation ever been studied through the lens of entropy? and seeing patterns and bifurcations as a mechanism to increase entropy?
So that's not that I know of in the developmental biology sense. And I think, I mean, it has been studied, obviously, in physical sense and structures and all like this. I don't know if that's been studied in the biological system. And I think one of the reasons for that is, again, we need to think about what is the system? I mean, is it trying to minimize something, given that cells are sort of energy producers and stuff like this? So it's not clear whether there's any sort of principle that, for instance, would be present in a physical system that should be present in a biological system, because of the fact that it's an open system, so it's generating energy due to having input from outside, rather than, say, a physical system where you might have a limited energy to look at.
So this is a question just following up. This is my own question. So there hasn't really been any interaction between Turing's work and that of the Brussels School, Prigogine and dissipative structures.
Oh no, so certainly Prigogine was one of the people who really pushed forward the idea. And in fact, I mean, having said that, I was thinking about it more from the biological point of view, but the chemical, I mean, those chemical reactions that I showed. Certainly, yeah, in that context, there has been studies of this. Yeah, absolutely. So thanks for bringing that up. I was thinking more in the biological context about whether it would be appropriate to talk about an entropy type of thing. Yeah, but you're absolutely right in the physical setup and in Prigogine, absolutely. Yeah.
All right, so let me return to that promised technical question. So Jim Swift asks, you mentioned advection briefly. Could you comment on the importance of advection of morphogens in pattern formation, which could dominate diffusion?
Yeah, so that I haven't looked at that. I mean, one thing that when we did some work where we had advection quite a long time ago, and of course one of the things that does is that then the Turing model, classical Turing model, gives you spatially are patterns that only change in space and not in time. When you start including things like advection, you can get oscillatory patterns, and so they may be of use. There may be, there's some situations where there are oscillatory patterns happening. I haven't seen any examples of where people have put advection as being the key dominating force for Turing stuff. I mean, for the stuff we've done later on, which is that Neural Crest stuff, their growth-induced advection seems to play an important role. People haven't really looked at that in the Turing context. And it's only been very recently that people have really even looked at growth in the Turing context. So I think there's still a lot of work to be done there.
Right, I think we probably have time for one more question. And I think I'll ask a question which is coming from Aaron Sloman. He asks, am I right in thinking that the mechanisms presented would not include explanations for forms of development that require particular chemicals to be produced in one place and transmitted to other places, or to control dispersion of waste products produced in one part of the system that need to be transported to where they can be discarded? Did you get that?
Yeah, so I think that, I mean, sort of thing that what these models are doing is they're sort of saying that we're looking at a particular area and we're asking, how do you get symmetry breaking in that area, and how do you get the structure to form in that area? What could happen in terms of this transport? And in fact, and this is known, for example, in the feather follicles that form on the back of the chicken, that there seems to be a propagation of a chemical, and as it propagates out from the backbone, that puts you into a Turing regime. It changes the parameter values, so you're in the Turing regime and you get patterns. So that could be an example of where, as something is transported along, pattern is forming behind it. But in terms of let's transport something to somewhere else, then what you'd have to do from the Turing point of view would say, well, let's just focus on where you've got to and see what the chemical does once it's got there, rather than worry about the transport of getting there. Yeah.
Right. So I'm being told by the coordinators that we are running short of time. So thank you, Philip. It was a really good discussion. Thank you, audience, for sending these really interesting questions. But I will have to hand things back over to the provost now.
Thank you. Thank you very much, Philip, for that wonderful lecture. Obviously, you had a tremendous number of questions. It's always good when the question session overruns. Thank you, Ronna Joy, for handling the questions. Thank you to our audience. Almost all of them staying with us the entire time, which is always a good thing, and for coming in the first place. And we hope many of you will come to the college soon when we can open up properly. And we'll share the date for the next Alan Turing lecture quite soon. Thank you. And goodbye from us. Thank you.
Thank you very much. Thank you. Thank you for the questions and the invitation. Thank you.